The HPG axis is the hypothalamo-pituitary gonad axis, and it's going to be what's regulating the hormonal control of reproduction. So in males, it's very straightforward. It's a regular negative feedback loop. So if we look at the structures, up here we have the hypothalamus. Down here we have the anterior pituitary. The hypothalamus has these nuclei that are releasing GnRH, or our gonadotropin releasing hormone. Gonadotropin releasing hormone will enter our median eminence and at the primary plexus be released through those portal veins then to the secondary plexus that are going to then pull around the cells called gonadotropes and stimulate the gonadotropes to release two hormones, FSH and LH. In just a minute, when we look at spermatogenesis, we'll talk more about the exact structures of the seminiferous tubules. But the cells that are in there that FSH is stimulating are cells called sustentacular cells. Some books just call them cystinocytes. Some books call them nurse cells. Some books call them, the other really popular one is Sertoli cells. And these cells are the cells that are going to nurture our developing spermatids. So they have the important job of, they're the nurse cells, so they're nursing the sperm. A couple other things that they do in males is they are going to produce this stuff called androgen binding protein, or ABP. And what androgen binding protein does is it's going to grab testosterone that's being released by the other cells we'll talk about in a second, and it's going to keep testosterone levels high in the testes because testosterone is necessary for spermatogenesis, or the production of sperm. If they didn't have androgen binding protein, then the testes would produce testosterone, it would go to the blood, be carried out of the testes, and we wouldn't have spermatogenesis. So androgen binding protein is going to bind testosterone and keep it in testes so that it's in higher levels in the testes so that it can stimulate spermatogenesis. Another thing that FSH is going to stimulate these sustentacular cells to do is to produce another hormone called inhibin. Inhibin's interesting. It sets up its own negative feedback loop on just FSH release. So inhibin is going to come back and it's going to selectively inhibit FSH release. And what this will do is it'll keep sperm count in homeostatic range so that if because these cells are really the cells that are nurturing our cells, and they're really helping with spermatogenesis. So if our sperm count gets too high, then inhibin can set up this negative feedback loop on FSH release, and we can stop sperm production while still keeping testosterone production up so that it stays in homeostatic range. So that's the FSH story. I don't have room on this board for LH2, so when you're all done, I'm going to erase it.
That says bind what and keep it?
Testosterone. We're good on FSH. Shout no, it's not, because I'm erasing. So the sustentacular cells are really doing a lot. They're nourishing the sperm as they're being developed, they're producing androgen binding protein, and they're setting up their own negative feedback loop. Alright, so the gonadotropes will also be stimulated to release LH, or the luteinizing hormone. LH is going to stimulate other cells in the testes called interstitial cells. These are are also called Leydig cells. And what these cells do is produce testosterone. And testosterone is going to stimulate spermatogenesis, which is the production of sperm. That's its primary function as far as reproduction is concerned. Testosterone also stimulates the development of secondary sex characteristics. So testosterone in males, we'll say, stimulates secondary sex characteristics. Another important thing that testosterone does for reproduction is stimulates libido, which is the drive for sex. In females, libido is driven by the adrenal androgen DHEA. So if you don't have a lot of DHEA as a female, you don't have a lot of libido. But males have a lot of libido because they've got a lot of testosterone. So testosterone is going to stimulate libido. It stimulates increased muscle anabolism, which is the buildup of muscle proteins. It stimulates increased bone growth. It stimulates thickening of the larynx and deepening of the voice. It stimulates increased facial hair and chest hair, axillary and pubic hair production. Pretty much it. So testosterone is pretty important. It's going to stimulate spermatogenesis, which is an important reproductive function, and then it carries out all these secondary functions for the development of our secondary sex characteristics in males.
Interstitial cells are also called Leydig cells?
Leydig cells, Leydig. That's a Y. And they're responding to LH to release testosterone. Any questions on the male HPG axis? So this is a normal negative feedback loop. When testosterone levels go high, it will feed back. So let's say this is high testosterone. It's going to feed back and cause a decrease in GnRH release. Let's say our testosterone goes low. It's going to feed back and increase GnRH release. So testosterone in the male reproductive system is a normal negative feedback loop. Females are non-normal. So that's why we're going to have 2 whole days to talk about the female system. Alright, so spermatogenesis is the production of sperm. And it's got 2 parts to it. The first part is called spermatogenesis. The second part is called spermiogenesis, but they're both part of spermatogenesis, so it's kind of weird. But the first part of spermatogenesis is if we look at a seminiferous tubule. This is a cross-section of a seminiferous tubule. This is the lumen. If you look in this seminiferous tubule, the bulk of the volume is filled by those nerve cells, the sustentacular cells. And so they're really big in there. So this would be a sustentacular cell. And if you look on the outside also has these germinal cells. Like in the female, our germinal cells were on the outside. So some books call those spermatogenic cells. Some books call them just germ cells. But what they are is the cells that are going to they're always going to be able to go under mitosis to give rise to new germ cells and other cells that are going to go into meiosis. So our germ cells are also called spermatogenic cells. And these divide by mitosis to give a lifetime supply of germ cells and sperm. So after puberty, males never stop producing sperm. So we'll say these divide by mitosis to give lifetime supply of sperm. That's the difference between males and females. Females are born with all the oocytes they'll ever have, and that the maturation of those oocytes stops at menopause. Boys are not born with any sperm cells. That starts at puberty, and then it continues through death. So our germ cells are kind of hanging out here, and they're dividing by mitosis to give rise to more germ cells, but then other cells that are going to become what are called spermatogonia. So if we look, then our spermatogonia are going to be the next cells that come down and develop into spermatids. So after mitosis, we have spermatogonia, or spermatids, and these cells undergo meiosis to divide their chromosome content in half. So that at the end of spermatogenesis, you have cells that are called haploid, or 1N. They contain half the number of chromosomes. So these would be haploid cells, or 1N. Spermatids, this is part of seminiferous tubules. So our spermatids undergo meiosis to divide the chromosome content. Our spermatogonia are going to divide and give our spermatids half the chromosome content. So as they're moving through here, they're going through these different stages of development, so that we get these little spermatids coming down to give rise to ultimately our sperm. So we're going to divide our chromosome content in half, and then we enter the next part of spermatogenesis, spermiogenesis. So our spermatogonia undergo meiosis. They're going to give us these spermatids. So the spermatids are, this next process is called spermiogenesis. And what happens in spermiogenesis is we take this spermatid now, and it's going to start growing a tail, or a flagellum. It's going to start developing this cap called an acrosome cap, and as spermiogenesis continues, then what's going to happen to give us a sperm is that we're going to lose all of this excess cytoplasm and really form this acrosome cap and grow this flagellum really long so that it can go off the epididymis and learn to swim. So in spermiogenesis, what we can say is our spermatids are shedding excess cytoplasm, they're growing their tails, and they're developing the acrosome cap. And what the acrosome cap is it's this protein cap that contains digestive enzymes. Those digestive enzymes are going to burrow into the oocyte. And one acrosome cap doesn't have enough digestive enzymes to do it. So it takes the coordinated effort of many sperm all trying to burrow in and releasing these digestive enzymes to really rupture that wall of the oocyte. Only one gets through, but it's lots of sperm in their acrosome caps that are able to weaken that wall. So that's spermiogenesis, when we take our spermatids and actually make it a sperm. So the spermatids have all this stuff that sperm don't need, and they don't have things that sperm do need. So they're going to shed off all the stuff that the sperm don't need, grow the things that the sperm do need, and then at the end of spermiogenesis, they'll be sperm. They're not reproductively capable yet. This all happens in the seminiferous tubules. The sperm, the late spermatids are released into the lumen. They shut off the rest of their stuff. And when they go to the epididymis and get motility, that's when they become reproductively capable. So you couldn't just pull sperm straight out of the testes and have it be reproductively viable, because it hasn't learned how to swim yet in the epididymis. Any questions on that? You can read about the details of meiosis in your book. I'm not going to test you on it. Usually you go over it in Bio 111. I know it's not a prereq anymore, so I don't know if you had it, but still there's just not enough time in this class. Alright, so semen is sperm and all of those accessory gland secretions. So it's sperm plus our seminal fluid. Again, our seminal fluid is going to nourish the sperm. It's going to help to neutralize the urethra and the female reproductive tract. It's going to clot the sperm together. Then it's also, semen also contains our prostatic fluid and bulbourethral fluid. So semen is released in the male sexual response in the second phase of the male sexual response. So we'll talk about that next. All right, for the male sexual response, there are 3 phases to the male sexual response. The first is the excitement phase. And during this time, thoughts of sex or mechanical stimuli are going to cause arousal in the male. The parasympathetic nervous system is going to take over blood flow in the penis, so that what happens is the blood vessels in the penis will vasodilate, and blood will come to the area, and then the veins get blocked from venous drainage, so it engorges the penis. So, and also during this time, the bulbourethral glands become active to start lubricating the penis. So we'll say thoughts of sex or physical stimuli will arouse the male. The parasympathetic nervous system is going to stimulate vasodilation and erectile tissue. Venous drainage is going to be inhibited. And what this is going to do is going to cause the erectile tissue to engorge with blood. This engorging with blood is going to cause the penis to become hard and increase in size. So we can say that the penis is going to increase in size and become erect. The thing that happens during this phase is that our bulbourethral glands become active and lubricate the penis and neutralize any, if there's any acidic urine hanging out, then it can neutralize that. The second phase of the male sexual response is ejaculation, and it's got 2 parts. So ejaculation is going to begin with emission, and emission happens when the accessory ducts are going to contract and start moving sperm. So sperm will move from the epididymis to the ductus deferens. The sperm that was in the ductus deferens will move into the pelvic cavity. So emission is the first phase, and this is when our accessory ducts contract to move sperm. Our accessory glands are also going to start secreting at this time to start adding their fluids to the sperm as they're passing by. So our sperm are going to move from the epididymis to the ductus deferens. The sperm that was in the ductus deferens is going to move into the pelvic cavity. Our accessory glands are going to begin secreting their fluids. And then the second phase of ejaculation is the expulsion stage. And what's going to happen during the expulsion stage is the internal urethral sphincter is going to contract so that no urine passes into the urethra. And our bulbous spongiosus muscle is going to contract, and that's going to forcefully expel semen from the body. Also, there are going to be rhythmic contractions of the muscle throughout the penis. So the second stage is expulsion, and during this time the internal urethral sphincter contracts. There's wave-like contraction of the muscles in the penis, and the bulbous spongiosus muscle contracts to forcefully expel semen from the male reproductive tract and deposit in the female reproductive tract. Second phase, ejaculation, is under sympathetic nervous system control. So the excitement phase is under parasympathetic nervous system control, and the ejaculation phase is under sympathetic nervous system control. The third phase in the male sexual response is called resolution. And during resolution, the the veins will be able to drain, the penis becomes flaccid, and during this time, males enter a refractory period when they are not supposed to be able to achieve another erection. This is a refractory period when penis becomes flaccid, and additional stimulation should not result in another erection. This phase increases with age, so the older you are, the longer the refractory period is.